Hello everyone, I'm Sherry, and today I'm going to talk constructing the relative importance of species sorting and dispersal limitation in shaping marine bacterial versus protist communities. To here, you may be questioned about what species sorting or what is dispersal limitation, or even you may be curious of that are they really do something in community assembly? Assembly means distribution and composition. Thanks to great advanced microbial biogeography, some people say distance-decay relationship can solve this problem. So you must know what is distance-decay relationship. It describes how the similarity of a biological community decreases as the geographic distance increases. So when you have a further distance, then you will get a lower similarity. So another question, what makes two closer places have a higher community similarity? I have to tell you, distance decay patterns suggest potential driving forces underlying community assembly. And the two major forces, one is species sorting and the other is dispersal limitation. Species sorting tells us the distribution and abundance related to the environmental or biotic conditions. And the dispersal limitation implies that not all suitable habitats can be occupied by species. It depends on the capacity to disperse to habitat. Let me explain you with the stories. When you zoom in these figures, then we assume there are three communities, and the distance between AB and AC are equal. But community A and C have similar environment factors, so the microbial tend to go there. This means species sorting. It just depends on what they choose, and the microbial will choose what it prefers. And on the other hand, there are the three communities. But no matter the environment factors, A and B have a closer distance, so the microbial can only swim to B. Then this is called dispersal limitation. It just depends on the ability to disperse. Then this is our aim, is to compare the relative importance of species sorting and dispersal limitation in structuring bacterial versus protist communities. So when we know that, then we can further to prove whether the two hypotheses which is more fit to our study. One is size plasticity. It means smaller organisms are less environmental filters than larger organisms. So that means it is more dominant by dispersal limitation. And the other is size dispersal. It tells us smaller organisms are more likely affected by species sorting than dispersal limitation. So it's more dominant by species sorting. And the small organisms in our study means bacteria. So to do this study, we first sample at East China Sea for 11 days in August 2014. And we have 33 stations for total 74 water samples. And we take the 3 water depths for surface and deep profile measurement and bottle. To see this photograph, and you can see the circle means surface, and the square means deep profile measurement, and the star means bottom. And the DCM have 2 colors. One is red and the other is blue. That means their surface and the profile measurement there are overlapping. So for sample preparation, we first get 15-liter seawater and then we filter it. Then we go to DNA extraction to get the microbial's DNA, and then go to PCR to amplify the DNA synthesis. Then we send to Illumina MySeq to have our DNA sequencing. And then we will, when the data sent back to us, and then we will go our program to do the analysis. And this is our data. You can see the reads. You can think the reads is how many individuals you collect. And then this individual ratio can separate into these number of group, then a station means a community. So this is just a part of the data. Then how do we study? First, we have to prove whether it is significant, means whether species sorting and dispersal limitation can really play a role in shaping community assembly. Then we use distance decay relationship, and for community similarity, we will use Gray and Curtis similarity index to calculate. This means the similarity equal 1 minus dissimilarity. And we will use distance-based redundance analysis to prove whether it is significant. Then we will do variation partitioning. It means how many percentage can be explained by the factors, and we separate into two factors. One is environmental factors, means it's more controlled by temperature, salinity, chlorophyll, or nutrient. And the other is special effect. It means it's more controlled by distance or dispersal ability. Then the region to circle are overlapping, means either can explain. So for E without S is pure environmental variation without a special component. It means species sorting. And for S without E, it means dispersal limitation. Then we get species sorting to divide dispersal limitation. This is so-called sorting dispersal effect ratio. Why we use ratio? Because it can distinguish the dominance of species sorting and dispersal limitation in a relative sense. So it's a relative influence. And we will use the new model analysis. It's called one-way ANOVA, and it's just a statistical way to calculate the sorting dispersal effect ratio. And then we will do habitat niche breeds. It's a crucial trait influence the relative influence of species sorting and dispersal limitation. And we will use this formula to calculate it. Then we will use GLMM and ANOVA to prove whether it is significant. So the most important thing is habitat niche breeds. When it's higher, then we know it's more governed by dispersal limitation. So what did we find? Look at these two figures. The x-axis means geography distance, and the y-axis means community similarity. The red means surface, and yellow means DCM, and blue means bottom. Then the point means the similarity between station 1, 1, 2, 1, 3, 1, 4, and 2, 1, 2, 3, 2, 4. Yes. So you will get these many multiple points. Then we use the distance-based redundance analysis. We can see that it is significant, means two factors are really exist. And next, when we look to these two figures, the x-axis it means the water level, and the y-axis means the species and dispersal effect ratio. And we can see in these two figures, the protists are triangle means protist and square means bacteria. And we can see in 3 layers and 2 figures, the protists are all higher than bacteria. This means protists were more governed by species sorting. And next, when we look to habitat niche breeds, we can also see, x-axis means two microbial, and the y-axis means the habitat niche breeds. And we can also see, and we can see bacteria are all higher than protists in these 3 figures. So this means bacterial communities are more governed by dispersal limitation. So finally, we got to discussion. So we find the lower sorting dispersal effect ratios for bacterial communities relative to protist communities in all 3 layers. This means protist communities were more governed by species sorting. And we also find bacterial communities are more governed by dispersal limitation. So it's the size plasticity hypothesis. The smaller organisms, in our study means bacteria, are less environmental filters than larger organisms. Thank you.
So, Tracy, Rich, Charlie, Roy, Jesse. Hey, wait a minute.
Previous slide. Yeah, here. So, can you explain more detail about why the higher habitat niche represent more governed by dispersal elimination?
Because when we have a higher habitat niche breeds, means means the microbial can suit or adapt to a wider environment. So when it can go everywhere they can go, then the environmental factors are not that important. It just depends on how many energy they can swim to that place. So that means it's more common by dispersal limitation.
Okay, Rich.
Can you go to the previous pages? Previous. This one. Can you explain more about why it's include sorting and the limiting about these pages?
What do you say? Why we separate into
Yes.
these two factors? Because when microbial, they tend to go another place, and it's when they first in A and then want to go to B, there are two factors to influence them to go anywhere, and the two factors. One is the environmental factors. They will be controlled by temperature, salinity. They will go to where they prefer to go, and the other is the distance. If the distance is too far, then they can't go there. So we separate into these two factors, and then we just take the place that they are not overlapping. The circle minus the overlapping regions, and that means our study species and dispersal limitation.
Charlie.
Can you go to a data page?
Data page.
Yes.
Okay.
Why the station named K in the 7 line, does it have any special means?
No, it's just a data, and K means the station's name. It's station K.
Roy.
The smaller organism is more likely to move twice. You say bacteria and the other one.
This?
Yes. It's about dispersal limitation. It's related to dispersal relation and dispersal limitation. The smaller organisms are less are more likely to move.
Can you repeat your question?
The small the organisms. The smaller organisms are more likely to move. It's because of dispersal limitation.
These two are two hypotheses. So either have people to stand for it, and some people say it's this. The smaller organisms are more governed by dispersal limitation, and some people say the smaller organisms are more governed by species sorting. So these are two hypotheses, and we just.
What is the difference between dispersal limitation and the size?
And the size.
Yes.
Dispersal limitation and the size.
Yes.
What's the difference between the?
Relationship.
You mean size dispersal relationship, or what? Limitation and size. It's just two hypotheses, and the hypothesis just say the smaller organisms. Yeah, and so you can 颠倒过来想 The larger organisms are less likely affected by species sorting.
Okay, I have to stop here. Volunteer question, Annie. Volunteer question.
You go to the data page. Why the lawyer, the SPR and DCN use the same station, but the button use the different station?
I just cut it random. It's just part of the data.
Any other question? No, okay. So, I think she gave a good talk, and when she presented her chosen paper, I told her that it won't be an easy paper, but I think she did a good job, so that I think you understood most of the concepts or ideas presented in this paper. And so the most critical, or let's think how she made it. And the key issue is she used a lot of charts to explain these concepts, like these cartoons. These are actually quite helpful. And also in terms of these variance explanation, it's also very helpful for you to understand how the authors did their research. So I think the flow of logic was really nice. And however, there are some things that can be improved. The first thing I noticed is that your font is too small. So when I was sitting there, sometimes it's not standing there. Some text was really small. So I think you have a lot of space for you to enlarge your font. And another thing is please avoid this kind of color coding, like text, white text over shallow. It's really poor. Where is it? Okay, this is really poor, so I can't see it at all. So please avoid this kind of color. And then another thing for you to improve next time is you really need to slow down. You were really fast. And because I'm not sure how many students in this class has ecology training. And even for me as a scientist, this is also new to me. So every time you say concepts, it always took me maybe 5 seconds to contemplate, okay, this is this, that is that. And so when you were really speaking like a blah, 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 then it's really difficult for us to digest everything. So you can slow down, and sometimes you can even pause. This kind of pause is not that you forgot your script. It's like you allow the audience to digest what you said. Okay. And then when you try, because this study use a lot of index, so these index are not straightforward, like it's not weight or length of fish or something. It's always based on complicated calculations. So whenever one presents things like this, it's always important. You did in some of the index, but not all. So for example, when you said index like this, you present this equation, which of course we will forget in 1 second. The most important thing is you need to say a higher, this similarity index means what? You always need to say that. Means higher similarity, right? It's, of course, but you need to say that, okay? Because sometimes it can be the other way around, so you don't know. And this one, when you were explaining this, you really need to slow down because you were extremely fast. And at some point, I started to forget, what does the circle mean? That means actually variation, but we already forgot. So you really need to slow down. Okay. And the font should be larger, you have space. Okay. And this, of course, you need to set a ratio value of 1 means what? Higher than 1 means what? Lower than 1 means what? Okay, so that's what you should always do when you present an index. Then there was things that, first, this is really small, and I don't know what do you mean by 2 factors here. So this seems to come out of nowhere. So you don't need to explain this time, but you should improve next time. So tell us what you want to say. Okay, but overall it's a good talk. So let's thank Sherry. Sorry, I have to use your break time because I feel I need to say something about, give me a second. Because some student came to me and asked, at the beginning of this class I said, it's possible for you to add subtitle and earn a bonus. Then one came to me and asked, do we add subtitle to the 3-minute video you sent us? No. Okay, that would be far too easy. Where is it? Okay. I didn't send separate email to inform the link of your Talk 1 video because there's a playlist. Here. So here you can see all your complete Talk 1 video and other students' talk. Sorry, I forgot